This is Alessandra Romano from University of Catania. Thanks IMF for the opportunity to share our results presented at the last ASH annual conference about the role of arginine deprivation in microenvironment to make multiple myeloma resistant to proteasome inhibition by metabolic rewiring. As several groups showed clearly at ASH this year, the progression of multiple myeloma is not only a genomic event related to the selection of progressive clonal that progressively acquire genomic events, but also it's associated to changes occurring in the microenvironment during a very important interplay between neoplastic plasma cells and environment cells. In this context, we are working on those soluble factors that can change the interplay between myeloma cells and the environment. In particular, we discovered that several amino acids, and one in particular, arginine, is progressively reduced in smoldering versus newly diagnosed versus relapsed refractory myeloma patients in the bone marrow and peripheral blood. But what is very important is this can occur in the bone marrow, where several cells are required to live together, and these cells are basically the myeloid suppressor derived cells, the high density neutrophils, which produce a lot amount of arginase, which is the enzyme devoted to degradation of arginine, and T cells that should fight against myeloma cells, recognizing them as neoplastic ones. But these cells are not able to synthesize or to produce arginine on their own selves. So for these reasons, they cannot be properly activated due to some changes occurring at the level of T cell receptors. So basically, our group clearly showed in the past that in the myeloma microenvironment, we have a very special situations for, devoting to reduce the amount of arginine. But what are the consequences of arginine low levels in the bone marrow environment for myeloma cells? What we found, very surprisingly, is that the availability of extracellular arginine is not detrimental for myeloma survival and immunoglobulin secretion. So it means that even if one amino acid is missing, myeloma cells are not worried about that and can continue to do their job. That means secreting the bad monoclonal component and proliferate. Even if in the proliferation there are some little changes, because we can have a delay in the cell cycle progression, but basically myeloma cells are alive. Just to be sure about that, we just to try to understand the molecular machinery, the molecular mechanism of myeloma adaptation. Surprisingly, we found that it's not related to autophagy induction, it's not related to GC2 activations, which are the responses that usually other cells use to respond to arginine deprivation. What we found is a new pathway, which is called eupmylation. Is not so different from ubiquitination. Probably we are more familiar to ubiquitination, which is the main target of proteasome inhibition and bortezomib, of course. What we found is that basically myeloma cells prefer to recycle the ribosomes, which are the organelles that are devoted to the synthesis of new RNA. And this is very smart to do because it can occur at early time points and consuming just a few ATP molecules. So it's very smart under the energetic point of view. And this is because during arginine deprivation, myeloma cells try to convey versus a low energy metabolic pathway, which means to stop to use glycolysis, stop to use mitochondria, try to save ATP. It's like when you have a lot of them, so you try to save money for what can happen in the future. It is exactly what myeloma cells do. And this is very important because it can convey a special adaptation pathway when you maintain in culture these cells for more than 10 days with low amount of arginine. What we found is the activation of inflammasome machinery, which can be a chronic inflammation. That means myeloma cells don't die, but they produce tons of interleukin 6, interleukin 18, and 1 beta that are required to continue to change the environment to support the cells. And this is very important because it's associated also to the very important, very dangerous attempt to rearrange DNA, because genomic instability can be acquired at that point to manage the high levels of reactive oxygen species and oxidative stress, conveying DNA damage, DNA leakage, and formation of micronuclei that in the past, and again, we showed to be connected to resistance to both bortezomib and carfilzomib. So to wrap up, what we showed is that basically arginine deprivation can induce a low energy metabolic state, poisoning myeloma cells into functional quiescent state. At the same time, this can convey DNA leakage and DNA damage that can further support the selection of new clones that are resistant to proteasome inhibitors. And at the same time, this can induce the activation of inflammasomes, promoting cytokine release that in turn can further affect the microenvironment composition and signals for the other cells. The implications of our work are pretty important, not only to understand new pathways that could be potentially triggered to overcome proteasome resistance to proteasome inhibitors, but it's really important also to use a target arginine to identify those niches in which it's easier to accumulate DNA damage. That is very important for us to prevent, or at least to identify those patients that can carry on DNA damage with the risk of developing subclonal events. So based on that, please let me thank all people in my lab that are working to, in this field, and let me thank International Myeloma Foundation for their support to go ahead with this field of research. Thanks a lot, all of you, for your attention.